my lab studies this group of organisms called segmented worms. And we typically look at how they regenerate. So I'll tell you a little bit about regeneration, and then we'll look at all the interesting things these animals do. And I am going to try to connect this to the emergent property phenomenon, which I think, as I thought about this, is worm. The worm is the emergent phenomenon here, I think, that these guys achieve through different ways. So I'll explain that. So as humans, we're not very good at regenerating, right? So if we lose body parts, if we lose an arm, if we lose even not maybe such a big piece of our body, but a part of an organ, we can't bring those tissues and organs and limbs back. And actually, even at the cellular level, we are not really good at, so let's just leave these complex structures, but just like cell types, we're not very good at doing that either, except for maybe that liver regenerates. But for example, with the success of childhood cancer treatment, even though now we're more and more successful in having these children become cancer-free, during treatment they lose their reproductive cells. And so that means that ultimately they may not be able to have their own children. So these are all examples of different levels of loss of tissue and cell types that we are not good at, but there are these animals that are very good at that. Right? So here's a segmented worm. So it was chopped into all these little pieces, and each of these little pieces will become a whole new worm. So they will not die. They will make a brain, head, tail, all kinds of different tissue types. And this is pretty widespread across organisms, and they do it in different ways. I'm guessing a lot of you know about sea stars being able to regenerate their arms, but actually here, a sea star, an entire sea star is regenerating from a single arm. So they can even bring back the entire body from a single arm. This is the original, let me see if this is going to work. Yeah, this is the original arm that's making a new sea star. And then there are animals that clone themselves, like Hydra. Hydra can make a little bud that ultimately turns into another Hydra that will detach and become its own. So all of these processes, regeneration and asexual reproduction through cloning, they require cells to coordinate, and genes to be activated. There are systemic signals like hormones, and it's a concerted effort, and it's very complex. And it's in fact so complex that one of the most brilliant scientists in biology, you may, if you're a little bit familiar with biology research, he's the person who established Drosophila as a genetic model. But Thomas Hunt Morgan actually initially started working on all these squishy, weird animals because he was interested in regeneration. So he actually did a lot of work on different animals, cut them and observed how they regenerate. And ultimately, he said, we will never understand the phenomena of development and regeneration. He gave up. He's like, this is just too much, too complex. And then went on to establish Drosophila fruit flies as a genetic model, won the Nobel Prize for it. So very good decision at the time, right? He recognized the limitations of technology at the time. But I today respectfully disagree with him. I think while he made a good decision, I think the wording of what he said was too sure and harsh. We will never understand all of these things. I don't think so. So I think it's a really exciting time in biology right now because actually really thanks to physics and a lot of other disciplines that feed into biology, we have amazing imaging technologies. We have other things that came through biology itself that allow us to look at these really complex phenomena in detail and with computational power, right? So I think we're now at the right time to look at this difficult question of how animals clone and regenerate themselves. So there are many animals that do this. You may know axolotl, the salamander on the corner. from memes and whatnot. If you're on the internet, they've become really popular. But basically what they do is they can regenerate arms. They can regenerate a bunch of other organs. We talked about sea stars. A lot of people know about lizards that can regenerate their tails. And this is a planarian worm, flatworm. This is actually what Thomas Hunt Morgan started with. And you can chop this little worm into tiny little cubes, and each of those little pieces will become a whole new worm. So they are really good at regenerating. So while these animals are all very good at regenerating, you may ask why my lab studies these ones, right? So we work on segmented worms, and these are the two species we primarily focus on. And maybe one thing I should explain here, what is a segmented worm? You know segmented worms actually pretty well. These guys are relatives of earthworms that come out after rain. If you're composting, the composting worms are segmented worms, and leeches are also segmented worms. I don't have any leeches in this talk, but leeches can be very cute. So if you want to see cute leeches, you're welcome to get in touch with me, and we can show you some at the lab. I recognize that these guys may look disgusting to a lot of people. We think, anyone that comes to my lab ultimately falls in love with these creepy little creatures. They are actually really cool, and I actually think they are cute. But I understand that you may find them disgusting.
What do they eat?
They eat all kinds of different things. So it depends on the species. There are more than 20,000 segmented worm species on Earth. They live on land, they live in the ocean, they live in rivers, and so it really depends on where they are. There are some carnivorous ones that eat other animals, but that's rare. It's more that they will eat sort of debris or algae, like large aqueous plants and stuff like that. Yeah, sometimes other smaller animals. Yeah. In the lab, we give them spirulina.
What?
And spirulina is this powder that people use for smoothies and whatnot. It's a powdered algae that has a lot of nutrients. So we just make them a little smoothie and give them spirulina. So we've got these little guys that are a little bit more modest looking, but actually segmented worms can look quite pretty. And if you want to see even prettier ones, I didn't put it here, but there's a Christmas tree worm. I highly recommend Googling that. And they come in different forms, but ultimately, the key thing about segmented worms is that they have segments. So they have these repeating body parts, and each repeating body part also has repeating nervous system. It's kind of like a sort of standard set of things in each body part repeated. But then depending on where you are along the worm, it may have some differences. Like the head segment has the brain, right? So there are some changes, but then there are all these repeated parts along the body. And then the other thing they do is they keep making more of these segments throughout their life. So they keep getting longer and longer. And we'll talk about that in a little bit. But we don't just study them because they can be beautiful and interesting. They actually serve a real purpose for us from a technical perspective. So we can live image these animals for days. And that's a really powerful method because we can actually watch as regeneration is happening, as maybe cells are moving to the cut site, to the wound site, right? And I told you about these other animals that regenerate, but the problem with them is if you put planarians under the microscope for too long live, they die. They just can't handle it because of their biology. And I think you can maybe appreciate if you've used the microscope how hard it may be to try to keep a lizard without moving for days, as it's in fact a lot of these animals take months to regenerate. So if we want to try to videotape, right, make a video of these processes, they are just not amenable to this kind of approach. And I always like saying that every organism that people have come up with for studying in the lab has its advantages and disadvantages, and basically you pick the ones that fit your purpose. So I'm really interested in microscopy and visualizing things live, and so these happen.
Do you MRI them or something like that?
Can you repeat that?
Use MRIs to,
MRI?
to image them, yeah.
You could certainly do that, and there are like some. So in the lab that I did my PhD at, we all studied mice and chicken. So there are like different parts of mice that actually regenerate a little bit, and so we had a machine that was made for that size of an animal to do. It wasn't MRI, but a similar thing. So we could visualize bone growth. The problem is if you want to visualize that continuously, because it takes a month for the bone to regrow. Again, you have to keep that mouse in that machine, or you have to bring it back, but position it all the same way, right? Like it's just very difficult. But certainly doable in like, sort of, you can do time points. You can image every day. Yeah. All right, great question. Okay, so yeah, so that's one of the primary reasons why we like these organisms. And they are, even though on the slides they look large, they are pretty small. So they are really easy to keep in the lab and put under microscope. And I'll show you some examples of this. So they are transparent, especially in this particular worm. You may be able to see this tubular thing. So that's its gut, which means that we're seeing through its skin, right? So it is so transparent that we are able to see its internal organs with just a light microscope, nothing fancy. And then they do some amazing things. So these are going to look a little creepy, but I think they deliver the message pretty well. So imagine that you lost your head, and you could just grow it back, no problem, right? That's what they do. So here is a video we've made. This is a worm whose head was cut off. And from here, it's growing an entire new head. And this is over 4 days. So we're able to keep this worm under the microscope for 4 days. And it's now, sorry about that, regenerating its little sensory organ. There is a brain that regenerates. Everything comes back. And it's a fully functional worm. What you're not seeing here, but we're able to also do, is to have more resolution in these videos and actually watch cells migrate, move towards the cut site, and their complex behavior. So it's really powerful. And it's just a worm without any sort of like dye or staining. Another thing they do, so again, imagine one day you started making more arms like this. That's what they do. So I told you that they add new segments. Basically, they do this from their growth zone over here at the tail end. So this is the tail, this is the head of this particular worm. And in this tail end, they have a set of stem cells that continuously divide and make new segments. So all the little smaller segments are newly made, and it's like a production line. And this worm will typically keep growing. Depending on the species, they may have a sort of limit, a number that they reach, and then they stop growing. But then there are some other worms that I'll show you next that do something with that very long body. They actually make more of themselves. So imagine that you started growing a whole new individual from the tip of your toes, and then they started detaching, and you made a bunch of clones. So that's what they do. So here's a worm, a segmented worm. This is a psyllid. This is the original worm, and then all of these little things are its clones. So here is the most progressed clone that's about to detach. And so it has tens of clones growing from its tail, and it's going to release these clones, and then they will become their own individuals, right? And it's all thanks to this zone at their tail end that has this stem cell population that keep producing these new body parts. So as if this isn't crazy enough, one more thing I'm going to show you that these guys do is this. So imagine you're making your clones, but they are branching out of you, and they are staying connected to you. So you're like this weird sort of superorganism. This was a worm that was discovered relatively recently. So here is, it's called a branching cellid because its clones actually don't detach. It just makes a lot of tails. And people thought, so this worm lives in a sponge. So here's a picture. It's not great, but you can kind of tell all these little white dashes, those are the tails that are sticking out of this sponge. When people observed these worms in the sponge and the worm in the wild, they thought these were multiple worm tails, individuals that were sticking out. But when they brought this to the lab and dissected, they found out that per sponge there is one worm. And it's all the branching tails that come out. So they have a mutualistic relationship. So they not only make clones, but they also kind of make this cool big branching version of themselves. So I guess at this point, as I was thinking about the emergent phenomena, I thought, okay, really this is achieving worm, meaning accurate body morphology after growth, reproduction, and regeneration. So what I want to discuss next is basically you go as a crazy scientist with an insult to the animal, right? So you may cut it in half, but somehow from these different cuts and different injuries, it's able to reconstruct a fully functioning worm. And it's also able to do this with these interesting growth and cloning processes, right? So it doesn't make bad copies of itself. It somehow, from wherever randomly it got cut or it starts making a new animal, it achieves worm.
Is this like sexual or asexual reproduction?
It's asexual. Yeah.
That's what I thought. So do they have both, or?
They have both, and in the same species, they can have both, basically. They can alternate between sexual and asexual reproduction, and some species only do one or the other, as far as we know, at least. Yeah. Okay, so essentially, in annelids, correct regenerated body plan is not explicitly encoded in any single gene, cell, or probably segment. Maybe segment, but no single cell in this animal's body knows this is what I'm supposed to do after an injury, right? They basically have to work together, so it emerges from these complex interactions, which is why I think, again, thinking about Thomas Hunt Morgan, it was just too complex that he didn't want to deal with it. And so after injury, for example, no individual cell knows how many segments it should build. Again, where is the head-tail axis? They may have some knowledge of that, but again, it's a lot of interactions between cells. Or when regeneration is complete, not an individual cell knows that, but I think there ends up ultimately being a systemic signal that delivers that message to all the cells. So yet the animal reliably restores the worm form, right? So I want to show you some examples of this. We talked about regeneration, so how they can make an entire new head, for example. But there's something else that happens during regeneration in the rest of the body. So even though for biologists for a long time, we've been really interested in like when you make a cut, what's growing back, right? Like that's really interesting. But there's actually a lot that goes on in the original part of the body. So we typically kind of ignore that because it doesn't seem as cool. But we actually think there's some really cool things going on in the rest of the body. And that connects back to this idea that the worm has to do something with the rest of the body too in order to have correct morphology. So I'll show you an example. So let's go back to this video. In this video, I told you it's growing an entire new head along with a brain. But in this original body part, there is also a lot happening. It's not visible in this video, but there's a lot of things happening in that part of the body. And there is a reason for that. So a couple of examples of this, both from regeneration and asexual reproduction, where the worms need to reconfigure are as follows. So here is an asexually reproducing worm. So basically this worm, this is the same species as we were just watching. It starts making a new head and a new tail in the middle of its body. And then these will split into two individuals. But the thing is, this part of the body used to be its intestine. And so now it needs to make it into a head. And then this worm that will become a new individual only has all the tail parts, right? It doesn't have the parts that are more towards the head. That I talk about it in terms of gut because it's the easiest. But basically, there is a very intensive reconfiguration going on here. It's not only making a new head, new tail, but then it has to reconfigure everything else. And there's a lot of plasticity here. So we look at this in terms of gut organization because it's really easy to observe. So these animals are actually quite similar to us. They have, even though they look very simple, they have all these different gut parts. So they have a stomach, they have different parts of the intestine, they have a crop, an esophagus. So this exists in the animal that we can use as a way to visualize this reconfiguration, remodeling process. So we can label specifically these different parts of the gut, and then we can ask, okay, now if this worm is dividing into two, or if I cut this worm, are these different parts of the gut coming back, and are they coming back correctly, right? So this is another example of a worm that was labeled for different parts of its gut. So we can merge that image all in one, but you can also see this marker, this only stains crop, this only stains stomach, etc. Right? So that's really nice to have because now we have a way to visualize these dynamic processes. These are fixed though, I should say. Like this is not live imaging. And then here is an example. If you go through the worm, you can see that blue and yellow, they don't overlap. They have a very sharp border. So it's really a precise labeling of these different gut regions. Okay, so, and then this shows that we have a bunch of different ways of staining different gut regions. Okay, so what's happening here, going back to our human as an example, is it's just something that these worms do specifically. When they lose their anterior part, head part, they just happen to only be able to, instead of completely making what's missing back, they only make a little part of the head back. So the problem here is this new individual is missing the torso part, right? Like it's missing everything in between. I actually want to ask you guys, what do you think, how can this organism solve this problem?
Can we keep going in the middle?
Yeah, so it could make something in the middle, like start growing things in the middle and then sort of squeeze it in, right? And then bring it back. It doesn't do that though. It's definitely a way to do it. Basically what it does is it takes takes this part and turns it into this part. It's this reconfiguration, active reconfiguration. And then it starts growing from its like toes in this case, right? Like it makes itself longer and then completes the rest of the missing parts. So it's an entire, again, reconfiguration process. Yes?
Are you saying the hydra reproduces the whole where the segments come?
So the worm basically what it does is it's using pre-existing parts and then changing them into the correct parts and then makes more cells more towards the tail, right? Is that what you're asking, or am I misunderstanding the question? Yeah. Maybe it'll make more sense in a minute with the following slides. So this is what the worm does. Not this. Okay. So in the case of the worm, we have these different gut regions that help us think about this question. We cut the worm here in the red dashed line. So just by its biology, this is what they do for whatever weird reason. When they regenerate their head, they only always regenerate 4 segments back. So they will only make this part back, right? So that puts the worm in a situation where now it's missing the stomach part. So it takes this intestine part and actively turns it into stomach. So it just is this like cellular reprogramming. It's not making more cells, it's not producing more cells, it's just turning what's pre-existing into the right, correct body region. And that's just sort of like a process that kind of goes through the entire worm. But because these are segmented worms, and I told you that they keep making new segments, this worm also grows longer and longer. So ultimately, it just reaches to a length long enough that it has all the body parts.
Yes?
So the first stage was to develop a head.
Yeah.
Okay.
Well, actually, I shouldn't say yes, because I'll show you next.
So it's one of the early stages. It develops the head, and then it like repurposes the other organs.
Yeah. So what we thought initially was that we thought the worm first makes the entire head and then does this reconfiguration, because when you look at just at the gross morphological level, that part didn't look like a stomach. But now that we have these molecular cell markers, and that's what I'll show you next, we actually found out that these two processes, the head growth and the intestine turning into stomach, occur simultaneously. And then we also did some experiments. I don't have that here, but when we inhibit head regeneration, the intestine still turns into stomach. So that's an uncoupled process, interestingly. Okay, so essentially, again, what's happening here is this. And so the way we showed this was actually through this. Jasmine was an undergrad in the lab, so she was a biology major. And she's right now a post-bac research assistant. We were talking about that. So Jasmine basically took worms and she cut them, like I told you in the cartoon, over here, where the anterior intestine is. So again, you'll see these colors. These are very specific for different gut regions, right? And so after you cut this worm, and if you stain those worms again for these, right after cutting them, they obviously will still have the anterior intestine. It's right after cutting. But if she waited for about a day and then did the staining, we could not detect the anterior intestine marker anymore, which means that those cells changed. They are no longer anterior intestine, but they are also not stomach yet, because we don't see any of the gut markers yet. So we think that this is actually a stage that these cells sort of pull themselves back to. They are not fully like stem cells, probably. We're investigating that. But they've lost their identity. So they are in this sort of more sort of plastic state. And now they're going to turn into what they're supposed to be. And so a day later, this is a little hard to see in the whole worm, but also, by the way, as you can see here, by day 2, there's a little thing that's starting to grow out, right? So that's the head. That's going to turn into the head. Here, when you look closely, you start seeing stomach cell stain coming back. So these cells are actually starting to turn into stomach. And then only a day later, so now the head is a little, head regeneration is a little bit more progressed, and we already have the stomach marker and this other region coming back. So it's all of those cells that used to be anterior intestine cells now are stomach cells. And it's happening simultaneously with head regeneration. And then if you go a little further, then you just get basically a worm that looks completely fine, except for actually some small differences that looks like this worm, but it came from this piece, right? So this I'm also not showing the entire worm to you, but basically it came from the more middle parts of the worm, and it turned itself into more two head parts. And this is a very fast process. It only takes 2 days. And so what we would like to do now is, because we can live image this process, we will do live imaging and see at the morphological level how these cells are turning into, from stomach cells into intestine cells. So that's one of the next steps that we have for this project. So it's a really unique opportunity, right? Like that we can visualize this entire process as it's happening in all of its complexity. And we can combine live imaging with these analyses where we can further look into what are the genes that are changing during this process. What is it that's driving this reprogramming, cellular reprogramming and remodeling? So that's what I have for this part. And maybe I can take a few questions if there's any questions about this. And then I want to tell you about the other species a little bit.
How's the stain? How's the stain work? Is it looking at which DNA, which genes are turned on and off?
Yeah, so this is.
So is it something that you're constantly adding to it?
No, so this is called in situ hybridization. So it looks for the, what's your background? I guess maybe, or it doesn't matter, so that I can make sure everyone understands. So every cell has the same set of DNA, same set of genes, but cells activate genes based on what they are, right? So my eyes have eye genes expressed active. So we can actually detect the mRNA, which is the active part of the, so what's called gene expression is that from the DNA you're producing RNA. So we can look for that. So depending on the cell type, it will have the same, everyone has the same genome, same DNA, but they have different genes expressed, meaning that they have different sets of mRNA. So that's what we are detecting here. And essentially we're using a gene that's specific for stomach. And then we're detecting that with a method that gives it a particular color. But it's fixed, right? So this isn't live. It is fixed at that point, and then we are visualizing at that time point whose stomach, whose intestine.
Okay, that's kind of what I was getting to. Is it when you're visualizing it, but you're only stopping it at a point.
Right, so in these colored ones, it is stopped. But then we can combine what we would find out from live imaging and sort of, one of the things we do is we live image an animal and then stop it, fix it, and stain it. So we can have that last time point, for example. But there are also other ways in this animal it's not possible, but you can actually make these stainings through genetic modification. And you can look at these things live in some other organisms. It's just technically...
Staining it once, and the animal's dead after that.
In this case, yes, but there are ways to do it in other organisms. It's just technically not possible with this one. Yes?
Do biologists see similar regeneration processes in plants? And if so, do you regard it as an evolutionary thing into the animals, or are they two separate paths?
So my speculation would be that regeneration evolved multiple times. So it's not all coming from the same sort of like ancestor. And that's, especially when it comes to plants, that's also because plants don't have cell migration. So because plant cells have cell walls, right? Like it's a very rigid organism in that sense. And they cannot, so what they do is they have a very good local response. Like when you cut a plant in that local spot, they have this plasticity and they can regrow an entire branch. But there is no cell coming from other parts of the plant to help the process. Whereas in animals, we have a lot of cell migration. So, and then we have this immune system cells that in a lot of animal regeneration, you have to first activate the immune system for these like cells to come and like clean up it's a mess, right? Like it's a bunch of cells that got like burst and cut and all that. So if you inhibit those early processes, actually regeneration doesn't progress. So I think there are commonalities, but there are differences. And even within animals, there are multiple points of regeneration, evolution of regeneration. But that's my speculation. It hasn't been tested yet, right? Yeah. You've been waiting, yes.
What is the hierarchy of the nervous system?
Maybe.
What
Is
do
it
you mean?
localized to the segment, or is there something that runs from segment to segment?
So
I I mean, they have a brain. they have a little, cute little, it's basically in the head, and it's a cluster of nerve cells. The brain has different parts that have different functions. And then the brain gets connected to a ventral nerve cord. So these animals, unlike us, like we have a spinal cord in our dorsal side, they have a ventral cord. And then that cord runs all along the body, and it sends, in each segment, sends these circular axons and projections. And in each segment, there is also groups of cells, ganglia. So, I mean, they have a nervous system that senses everything along the body. We anesthetize them before cutting them because we know they feel pain. At least that's philosophical. We don't know the type of pain they feel compared to us, but yeah, they have interesting behavior. I'll show you actually in a minute. They've got some pretty complex mating behavior in some organisms. Yeah, so. And nervous system is known to be important for regeneration as well, actually. So people have done these really neat experiments before they had all the fancy techniques that we now have. They did these things with the animals. So they would take a part of the nerve cord and then deviate it, so instead of it going straight, it would go this way, and you actually get a worm growing, tail growing from here. Because it's inducing that in that location. Yeah. You had a question?
Yeah, could you excuse me, starting from an individual cell, what can you get? Did you say you can get something that looks like a worm, but it's...
In this worm, just a single cell can't remake a whole worm, right? But it cannot. Just one cell, I mean, unless it's a fertilized egg, right? Then it can, but in regeneration, it cannot. There are, interestingly, species-specific limitations. So this worm will regenerate from 3 segments, fine. There are some worms that can be as small as 1 segment. So it's really, you need to kind of test it based on the animal. In Hydra, which is a jellyfish, Cnidarian, it's a different group of animals. You can actually split Hydra into its individual cells, and as long as it has a number of each cell type, even if it's small, you make a little aggregate, a little ball of cells. Those turn into a Hydra. So, but these guys can't do that. So it's different depending on the species.
Yeah.
So how many?
Yes?
Are there any diseases that they can't regenerate from?
They are actually really hard to sort of make sick. So unless we don't change the water and let them in really bad conditions, or if you step on them, that's kind of too much. But we haven't really tried making them sick. So yeah, that's a great question. I'm gonna think about that.
Yes?
So how many nerve cells per segment? I mean, C. elegans has a famous 302 cells, so nerve cells.
This is definitely an answerable question, but I don't have the answer myself.
Well, the other question is do they have a circulatory system?
They have a circulatory system, so they have blood vessels.
They have blood vessels?
They do, yeah,
Okay.
yeah.
Yeah, so they are not like insects, right? Where there's an open system where everything kind of just mixes up, and so this is actually, they don't have a heart, but they have these heart-like thicker and more stronger muscular vessels towards the head that kind of almost do the pumping.
Okay.
Yeah, and you can see the blood flow. Yeah.
Okay.
Yeah.
Yes?
Where are the blood cells made?
Oh.
Actually, there's a paper about this, so it's. Oh yeah, there is no bone, of course. Yes, I mean, so I don't know too much about the blood system, but there's this one paper that was published a few years ago, and what I remember is they do have hemoglobin, kind of carrying cells And then those are made in some like pockets of like segments, but I don't remember. Like there is a particular region, but it's not, obviously it's not bone marrow.
Each segment?
In each segment, yeah. So they showed like this, these little pockets that where these cells were being produced, yeah. Yes?
Maybe I missed this, but can we say precisely what humans are missing that precludes humans from doing what these segmented forms do? And is the research thinking that maybe someday we'll figure out how to transfer that regeneration process into humans so that the childhood cancer patient can reproduce?
So
we don't know, right? That's the big question. Essentially, we really want to understand what is it that they have that we don't. Especially knowing that we share, I mean, with these animals, we share probably, I don't know, something like 70% of our genes, right? Like there's a lot of similarities in that sense, but clearly there are differences. And so that's the big question. And from an intellectual perspective, for me, I would of course love for my research to help humans. But to be honest, I'm just really curious, like how are they doing this, right? So, I mean, hopefully that will help us develop ways of helping ourselves. And I think that it will ultimately, because really understanding how a cell is like getting some sort of signal and going back to a more like sort of clean slate state while in the body, right? Like it's not in cell culture dish. And then it's still keeping the gut integrity too, right? Like it's not just dissolving, like these organs aren't just dissolving. They're still keeping their integrity, but they are reprogramming ourselves. I think there's a lot to learn from that I would like to be hopeful that it'll help humans, but I am also just purely curious about how are they doing this, yeah. But I think I should also point out that some of these comparisons are better made between more closely related species. So one of the things that we're interested in pursuing in the future is there are, so this species has a sister species. It's a very evolutionarily closely related species, but that species can't regenerate its head. So that's actually a much easier comparison because like worm, a lot is similar, only recently diverged from each other. What's changed, right? So that's actually, we're actively working on a grant with some other people who work on different organisms to make these kinds of comparisons. So I think those are going to give us some really good answers. Okay, so I want to, with the time remaining, I want to show you a little bit this other species. I think it's just fun to also look at what it does. This is, let's see. This other species we study, unlike the one that I've been showing you that splits into 2 or 3, these guys don't do that. So these guys don't reproduce asexually. They reproduce sexually. So you're seeing here a female at the top and a male at the bottom. And we use this particular species to try to understand how they regenerate their reproductive cells. Again, for a bunch of technical reasons I don't want to bore you with, they are much better suited for that question. Something that's interesting about these guys, which is what I want to share you with, is as juveniles, so like I guess teenagers, they look all similar to each other. So whether it's a female or a male, you can't really look at the worm and tell whether it's a boy worm or girl worm. So one of the things that we have done so far is to try to find ways to be able to tell that, so that when we're making these experiments for reproductive cell regeneration, we can pick we can sort of investigate this based on also whether they are a male or female, right? So how does this worm on the right turn into these guys on the left, right? So it actually undergoes an extensive metamorphosis in order to fully mature and be ready to reproduce. It loses the entire gut. So in here, all the sort of brownish thing you see is its gut. again, it's food/poop. That gut is not present here, as you can tell. Oh, by the way, someone was asking about blood, right? So this is the blood vessel that's running across this worm. But anyway, so this drastic metamorphosis generates the mature worms. They also have larger eyes. But as you can imagine, without your internal organs, what are you going to do? They actually don't survive much once they mature. So they have a very dramatic life cycle. They spend most of their lives as juvenile worms, teenagers. And then, or maybe that's not a correct analogy, children. And then suddenly they mature over the course of a couple of days. And then they mate and they die. So that's just like this dramatic life cycle. So I'm going to show you how they do the mating. We can take the male and female worm and put them into a dish. And they do this thing called a nuptial dance. So they exchange pheromones with each other. And basically they kind of like, I call it consent. They're like, is this going to happen? Yes, this is going to happen. And then they both release their gametes into the water. And ultimately, this is, for especially the female, it's a pretty destructive process because the eggs just come out of the sides of her body. And then ultimately there's nothing else left. And the male survives maybe for a couple more hours, and then they both die. And upon that, you end up with all of the little dots in the dish are eggs that will become cute little worm larvae. And we can take those eggs, inject them with things to change their genome and be able to visualize things. So here are some eggs that were injected with a dye. And actually, in this case, you were asking about this. So with this worm, we can visualize live these stained cells because we can inject their eggs. And so here, its nervous system is actually in the middle, visible. And this is a live worm that we imaged. So again, depending on the worm, you can do different things. But then the other thing I want to tell you here is so I guess maybe one thing to point at here is if you're a worm and you have only one shot at reproduction because you mature and you have only one day to find another worm and mate. From an evolutionary perspective, there's a lot of pressure on finding a mate because if you don't find it, you're dead. Like your gene pool, your genes aren't getting passed on, right? In order to be able to find each other, these guys evolved synchronizing their maturation. So they synchronize when they mature with the moonlight. The moonlight causes the brain cells to start releasing a hormone that then tells the rest of the body it's time to mature. And so over the course of 2 weeks, the worms will mature and they will be able to find each other. Any worm that matures out of schedule gets selected against. Like it doesn't contribute to the gene pool. So it's a really strong evolutionary pressure. And there are actually a lot of examples of this in the wild with other organisms too, because they need to synchronize. And there's a lab in Vienna that studies this. They've removed the genes that are important from the eyes to perceive the moonlight. And when you remove those genes, those worms still mature, but they don't synchronize anymore. They just mature all across the month. Yeah, so that's something really interesting about them.
Yeah. So how do you stimulate moonlight in a lab environment?
Oh yeah, great question. How do we simulate moonlight in the lab? It's a very cheap little LED light. We have a timer. So we don't really emulate the moon phases. They don't really need that. The moonlight comes on about 10 days every month. And then otherwise, it's pitch dark in the culture room. Yeah. So it's just on a timer. And then we keep them always in the summertime. So that means they have 18 hours of daylight and then 6 hours of night.
Are they being affected by light pollution?
So this is a question I asked myself quite a bit because in the wild, right, so you can find a species similar to this on the East Coast here in the US, and I've collected them. Somehow they still were maturing on schedule. So maybe maybe the lights that we have aren't strong enough. I'm not sure. I think they are not yet, at least in the area. So this was Cape Cod. You know, it's maybe still not that light polluted. There's a lot of like nature around and all that. But I would say that certainly in the lab, if we have an extended period of, like if the lights are wrong, things shift for sure. So yeah, I'm not sure. Maybe in the wild, because they live at the bottom of the ocean and they live among the leaves. I mean, yeah, I don't know. This is something that definitely is in my mind that I don't have an answer to. Yeah.
Yes.
What percentage of those gametes form another animal?
They are, if this is in the wild, they are, or like if it's a healthy couple, it's typically almost 100%, but they get eaten. So it's a number game, right? Like there's always, so when I collected them, we normally culture them in the lab right now, but when I collected them in the wild, there's always fish around because it's like nutritious, egg rich, you know, like so they are not only maybe getting eaten as themselves, but their progeny gets eaten too. So I think in the lab sometimes we have less success. I think that's part because we kind of force them to choose well, they don't have a choice, but maybe like it's not as good as a male, but we still sort of pair them up. But it's pretty good percentage, yeah.
What depth are these at?
They, so you can find them on the shore. They're not like deep water species.
So it's more like tidal pools?
Not tidal pools though, so they like making these tubes. They build tubes on large algae or rocks. So it still needs to be a little more than like just like the shore, but you could go like with a wader and like collect rocks or algae. Or if you're ever, you know, on the East Coast during the summer, you can look up the moon calendar online and then sort of go about 10 days after the moon. You can get in touch with me and you can be like, hey, Duygu, I'm on the East Coast and I want to find the worms. I'm happy to help you think about the timing. And then you just go at night with a good flashlight and then they will come to the surface. So this mating happens at the ocean surface, but they normally live at the bottom. Yeah. Yeah. Yeah, and we, like I said, we have them at the lab. I would actually love to do like a live demo at some point, but it's a little complicated with their schedule. So let me see. I have only one more thing to tell you. So this is with their reproductive cell regeneration, right? So they are really, again, weird in that their reproductive cells are sort of spread across their body. There's no organ that hosts them. So if you cut this worm, it will regenerate its tail, and then along with that tail regeneration, it will regenerate its reproductive cells. And we're just interested in that particularly because that affects the next generation, right? So one, what are the genes involved in this process? How does age affect this regeneration process, the age of the worm? And then are the regenerated reproductive cells as healthy as the original ones? So if this worm is making babies from regenerated reproductive cells, do those babies have more mutations or are they fine? Which we actually have indication that they are fine, so they must have a way of controlling the genomic integrity, the mutation rate. So that's those are some of the other questions that we're interested in. I just want to make sure that I thank my lab and funding sources and collaborators, and I'm happy to take more questions. Thank you.